Hello, welcome to chapter 21. This is skin and eye infections. We're going to start with anatomy of the skin and anatomy of the eye. The skin you've covered before in our innate defenses, and you should remember that our skin is a really great barrier for microbes of all kinds because the top layer is very tough to get through for a lot of microbes. It's not a very welcoming environment for microbes to live on because it's basically very dry. Let's just go through the terms here, right? So the epidermis, which is the very top of the skin, the super top, like the one that we touch, is actually just dead cells. So this very surface will be dead keratinized skin cells. It's going to be dry, they're salty from our sweat, it's cool relatively to the inside of our body, and it sloughs off every day. It's also acidic, right? Set up by the normal flora and by the sebum of our sebaceous glands, and it sloughs off every day. So if you had some transient bacteria there or pathogens, it would fall off eventually with your skin. However, underneath our very top dead layer, right, we have living cells, and they replace the upper cells, and they contain our phagocytes, like for specific example, our dendritic cells. So in this histology slide of skin, tissue slide, you can see that the epidermis is everything that's dark. So it's relatively thin compared to the really big area we see here, which is dermis. If you look at here, right, we have a very thin epidermis and a really thick dermis. So the dermis is where you're going to have your blood supply. The epidermis is very top layer. It does not have a direct blood supply. So for microbes that inhabit here, they have to actually penetrate through many layers of cells before they get to the dermis where the blood supply is. And so another thing that you can think of is that when you scratch yourself or you bump into something and you can see that the skin has come off a little bit, but you're not bleeding. That's because you've only cut off or like rubbed off that epidermis and not hit the dermis yet. So the dermis is a more livable place for microbes. You can think about that because it does have a blood supply and there's nutrients present. It's a warmer, moister environment. The primary stuff that's there, right? It's not a bunch of cells. You absolutely have cells, but it's a lot of connective tissue, especially the collagen, and different, proteins that we've heard about before because bacteria have evolved enzymes to degrade them. So if you remember, like collagenase can degrade collagen, and we have hyaluronic acid, and bacteria have hyaluronidase. So it's quite protein rich in there. We have our nerves in our dermis. We have our sweat glands and oil glands in our dermis, and we also have our hair follicles. So lots of stuff in the dermis, right? And then the hypodermis, this here is all going to be predominantly fat. The hypodermis is fat tissue, but it also contains a blood supply. All right, so this is just a nice picture of your very top layer of skin that is dead. So your cells, the purple ones, would be completely flat like pancakes, and they would be filled with a hard protein called keratin. And the picture here shows you the different kinds of microbes they can find in your skin. And there's the picture of it. So we have lots of different bacteria. What you can't see are, any viruses that might be present because they're too small to get picked up. So there you go. So when we get to our normal flora, you can see from this chart that there's lots of different bacteria and other kinds of microbes on the skin. This one's just bacteria. And there's going to be a difference of the population due to the environments of our skin. So we're going to have dry regions versus moist regions. We're going to have warmer regions and cooler regions. For example, the tip of your nose or the entrance of your nose is going to be cool compared to maybe like the armpit area, right, which is pretty warm. So there is a significant variation in our skin. And remember that it's not just bacteria. We have fungi that live in our skin and also viruses. The most abundant from here you can see is Staphylococcus. So we worked with Staphylococcus in our lab a lot. And then Corynebacteria. This is the genus term for that. And we find those widely spread throughout our skin. Oops. All right. So when we get a skin infection, it's due to several ways, right? Sometimes we actually carry the pathogenic normal flora. So one in note is MRSA, which stands for methicillin resistant Staphylococcus aureus. And if one person has it as normal flora, they're not bothered by it. Of course, it can become pathogenic if it goes deeper into the skin, but some people just live with it on their skin. However, if nobody knows that they're a carrier of MRSA, but let's say that you are working in a hospital, you're a carrier of MRSA, you could possibly unwittingly pass it on to someone who's sick, immunocompromised, has some kind of open wound. And so you could pass the disease on to people that are actually going to be more susceptible. Sometimes you have injury to the skin, right? And bacteria can go in deeper into deeper areas of the body. So it can be normal flora, or it can be some other pathogenic bacteria. And also the hair cells, so our hair follicles and sweat glands. So those are going to be environments that are going to harbor a lot of bacteria. And if they get there, it can be a local infection, right? So it could just stay in the hair follicle or stay in the sweat gland, or because we're talking about dermis, right? And we have a blood supply there. When it gets into the dermis or the hair or sweat glands, it can spread to the blood. And this is true for anything that goes into the dermis, because the dermis has the blood supply. So those deeper infections are the ones you kind of want to worry about, because they could possibly pass into the bloodstream. So we're going to be talking about these guys: Staphylococcus aureus infections, Streptococcus pyogenes, Pseudomonas, and Bacillus anthracis, and then of course we have acne. Before we get to the actual diseases, these are some terms that you will come across in your reading. So this is a nice chart of the things you should be familiar with. I'm going to let you guys go through this yourselves. I don't need to read it to you, but there's lots and lots of terms for different kinds of bumps, and whether it has pus or fluid, whether it is a flat area or if it's like deep. So I'll let you guys read that. This is just an image of those terms, so it's nice to have an image. Wheals are something you probably not heard of, but these are going to be things like when you get an insect bite or a mosquito bite and it itches, that's a wheal. I'd like to also point out that a cyst is going to be completely encapsulated. So if you have a cyst of some sort, I know sebaceous cysts are quite common. They can actually just take it out as a bag, because it does have that membrane around it. So it's all contained within a little bag. And so sometimes people get cysts removed, and it's quite interesting. They just take out that whole bag of sebum usually. All right, for the eye, we have this lacrimal apparatus. So a callback to anatomy. This is the gland, lacrimal gland, that actually makes the tears. When the tears are produced, they're going to travel through little ducts, and they go into the eye from the upper outer part of the eye. Then they're going to wash over your eye, and the tears are going to drain into two little tiny holes that you may have seen in the mirror. Those are called puncta. So just think of it like a puncture. And then there's more ducts that lead to a sac, and that sac will take those tears and drain them into the nose. So ultimately, the tears that you make will end up in your nose. And tears are really great. They lubricate the eye, obviously, but they also contain many antimicrobial substances, such as lysozyme. You should be familiar with this. We talked about this enzyme before, and it breaks down peptidoglycan in bacteria. There's also something called defensins that disrupt the membrane and the wall of bacteria. There is this flushing action, right, that washes away our microbes. And then there's mucins that also trap and promote the removal of microbes. So tears are really great innate feature of our defenses. The eye, okay, review for another anatomy lesson here. So the eye has a very hard outer covering, which is called on the front of the eye, the clear part's called the cornea. And then the white of your eye, everything from here back, that's going to be called the sclera. I don't think it's mentioned here, and you don't need to know that because it's an anatomy sort of thing. But know that the eye is not expandable at all. So any pressure, any extra fluid or bleeding in the eye can actually compress the delicate cells of the eye itself, and that can lead to all kinds of problems. It can also lead to blindness. But we're going to talk a lot about the conjunctiva. Okay, so the conjunctiva is a membrane. It's basically a mucous membrane that covers the white of the eye. It stops before you get to the clear cornea because we don't want the light to get stopped by that membrane. Picks up again here. It actually coats the eye until you get to where it stops, and then it does a U-turn and it comes and it coats the inside of your eyelid. Same thing down here. The conjunctiva, this membrane, will protect the eye, and then when the eyelid stops, it'll make a U-turn and then coat the bottom of the eyelid. So there is, unless you break this conjunctiva, things will not go to the back of the eye. So if you get something in your eye, it might be lost under your upper eyelid, it might get lost under your lower eyelid, but there is a blind ending to this. So things like maybe you get your contact lens lost in your eye, like it's never going to get lost in the back of your eye socket because you have this barrier. So that's something that the conjunctiva does, is it provides that nice barrier to things that it might, want to travel deeper. It does contain blood vessels. And that's why when you get irritated in the eye, your eye becomes red. And notice that it's only the conjunctiva, right? The membrane that becomes red, and not the cornea. The cornea is going to always stay clear. Now, what is the cornea? The cornea is going to be made out of collagen. So our tough connective tissue. And there's a really thin layer of cells, which is kind of surprising because we think that, we're looking at the world and it's clear. But there are cells there and they can regenerate the collagen. So it is renewable. So if you scratch your cornea, you damage your cornea, it can be regrown. And normally we would say the inside is sterile, right? The inside of your eye, all this stuff here is free of pathogen. Outside the eye may contain some flora, but most of all, it's pretty clean. The tears wash away a lot of the bacteria. Infections of the eye can then be of the conjunctiva. So just that membrane is going to get inflamed and red. So you can see here the conjunctivitis. So the conjunctiva, you can see where the iris stops, right? So that's where the cornea stops, and everything else that's red is going to be that mucous membrane. Blepharitis is of the eyelids themselves, not the eye. And then keratitis is an inflammation of the cornea. So you can see here, obviously there's going to be irritation in the mucous membrane as well, but you can see the cloudiness, how it builds up in the clear part of your eye. That would be keratitis. All right, so we're going to start off with Staph aureus. So Staphylococcus aureus is a very prevalent pathogen and also normal flora of many people. It is contagious from person to person, direct contact and indirect contact. So indirect meaning that something like fomites, right? There are many drug-resistant forms. The one that we've heard of the most is MRSA, and it's still an issue for hospitals all around. And then why Staphylococcus aureus is such a pathogen and such a big deal for the health community is because it has all of these different substances that can make it very, very hard to kill or, cause disease that's pretty severe. So first off, we have a group of toxins called staphylolysins. Staphylolysins, there's several of them. Alpha toxin is the one you want to remember because it's also mentioned in lab. So alpha toxin comes up in your lab final as well. Alpha toxin, what it does is it produces pores in the cells and it lyses your cells. So an example of alpha toxin that we did in lab was when we inoculated the blood plate. Remember, the blood plate can be differential for what kind of reaction occurs, whether the bacteria can completely break down the red blood cells or just partially break down the red blood cells. So alpha toxin will completely lyse the red blood cells and produce a clear area on the blood plate. Coagulase is an enzyme, and you know this by ending in ASE. This can form abscesses. So coagulase is going to be able to coagulate the proteins in our fluids, right, and also in our blood. And your body will coagulate substances to kind of contain them. But the bacteria can actually make this themselves, and so they can hide inside of this coagulated region. And then that in turn will form an abscess because you have an area that's walled off. The bacteria are going to be dividing in here, right? Through a face. Bacteria will be dividing in there, and it'll form basically an infection within a swallowed off area. Leukocytin, leuko, you can remember this as a leukocyte, white blood cell. These kill white blood cells, right? And so when you have white blood cells that are coming to an infection, there's pus created, there's inflammation, and when the white blood cells are killed, it leads to pus. Lastly, there's something called protein A that attaches to the constant region of antibodies. So protein A, I thought needed a visual because we can see that this is SA, this is Staph aureus. We can see that this is protein A. So basically our antibodies, right? Our body will make these antibodies. And in normal conditions, the antibody would bind in this other way. It would bind the variable region, right, to the antigen. And then this constant region, remember that was the one that macrophages could hold on to. They could, it helps, remember the role of antibodies was to help opsonization. But what this protein A does is it can actually flip it. It attaches to the constant region, and therefore it makes it really difficult. Like you're basically taking that tool away in this picture here, right? This is going to be a neutrophil. It has a receptor for that constant region, Fc receptor. So if it were facing the correct way, then you can see that the attachment site is available and you can get phagocytosis really readily. But when you take it away, then we have decreased phagocytosis. So lots of tricks for Staphylococcus aureus they have. And this is one of our staph infections. This is going to be a superficial. So we're only talking, when we say superficial, we're talking about, epidermis and very shallow dermis. So we have folliculitis. This is a picture of folliculitis, which you can imagine is infection of the hair follicles. They're going to present as bumps and pimples, and actually acne is a form of folliculitis because that bacteria gets into your hair follicles. And the causes of folliculitis can be many, right? Sometimes sweat buildup can just clog those pores and trap the bacteria inside. Injuries, ingrown hairs, sometimes wearing tight clothing can do it, or any irritation. Sometimes shaving can do that, right? So I think we've all probably experienced some folliculitis in our life. A boil and a carbuncle, probably less common. Boils are going to be deeper. This is a boil. It is going to be more severe, obviously. And this is going to have more of an impact when you get it, right? So this is stuff that is everyday life things that you can't really avoid. But boils are more with like athletics, sports, athletic equipment, or you have a weakened immune system, such as poor nutrition. And so your body just can't fight off the bacteria. Remember, staph is contagious. So if you live in close quarters, boils can be contagious. A carbuncle is just several boils that come together and they form a deeper lesion. And so the deeper you go, remember the possibility of getting to the blood is more. So we would worry about systemic infections because it's a very large infection of the skin. And all of these can be prevented with proper hygiene. So this is a bigger, just a closer look at folliculitis. And this is acne. As I said, acne is a form of folliculitis. Yeah, so that is it. So for acne, the inflammation that's created and forms that folliculitis is basically because your hair follicles, there's inflammation present, right? Because you have a bacterial infection. So you have inflammation, and then they can become clogged with the lipids that you have there, like sebaceous gland, sebum, the fatty acids, and dead skin cells. And these are all really good nutrients for bacteria, right? They can eat the fat, and those dead skin cells provide protein, and you have this warm environment. So this is impetigo. So this is a condition of, now you're going to have some vesicles. Vesicles have fluid, okay? The difference between vesicles and pustules. Fluid. That's usually clear, okay, and watery. Pustules, it's be more like pus, right? So it's going to have yellowish, thicker exudate. or bullae that rupture, and they produce these encrusted sores. So one of the hallmarks of impetigo is encrusted sores, and the crust is pretty yellow. I guess I can say yellow, and it's often accompanied with seeping. The exudate, the fluid that's coming from those lesions are going to produce fluid, and then they're going to dry around the sore, and then that creates that crust. So the funny thing about impetigo is that it does occur normally around the nose and mouth, because that is where we have most of our Staphylococcus aureus that causes impetigo. Impetigo is common in kids, and it is also highly contagious. So if you have kids later, or if you have them now, be aware that it is contagious. And this is one form of impetigo. You can see that there's 2 bacteria that are the most prevalent causes of impetigo. So we have Staph aureus and we have Streptococcus pyogenes. Those are the 2 we're talking about. Or you can be unlucky and have a co-infection of both of them. Okay, so they're treated with antibiotics. It clears up without scarring. All right, so Staph aureus, how would you want to diagnose it's a Staph aureus infection versus maybe a Strep pyogenes infection? And this goes back to lab information as well. So gram staining, right? We know that Staphylococcus is a gram-positive coccus that grows in clumps, right? That's our staphylo right there. The MSA plate is also used to determine whether it's Staph aureus or not. Remember that MSA, the M stands for mannitol. And we have a red area and a yellow area, and you should know this by now. The yellow area is where mannitol was fermented, and so the acids from fermentation turn the plate yellow. On this side, we have another species that can grow but does not digest the mannitol. Another thing about mannitol, if you remember from lab, is that the S stands for salt, and both of our Staphylococcus or our Staphylococcus species are able to live with a high salt concentration. So this is a plate that's going to select for salt-loving bacteria or halophiles, and it's also going to differentiate based on mannitol fermentation. So we would look for a positive sign for mannitol fermentation for Staph aureus. Blood agar, you see here from lab, we have what's called beta hemolysis, right? Beta hemolysis, which is the complete breakdown of the red blood cells. And so we can see that there's a clear zone, right? We can see the colony in the center, and then we can see that clear zone around the colony, which means that the red blood cells around that colony have been broken down and are no longer present, so it doesn't look red anymore. The last 2 tests, coagulase, positive for Staph aureus, okay? So again, coagulase is an enzyme that can form clumps of the connective proteins that are found in your blood. And so if you put Staph aureus into a test that contains those proteins, it'll coagulate them and form basically a clump of those proteins, I guess. And then the negative, you would not find that. Coagulase net positive, and then catalase also positive. Catalase positive. So we remember the catalase test from lab. All right, so our next one is going to be Streptococcus pyogenes. The virulence factors are really similar to Staph aureus. They're different names, but notice that we have streptolysins, whereas Staph aureus had staphylolysins. I'm sorry, it was called staphylotoxin. Anyhow, but these are toxins. So O and S are 2 exotoxins. Remember, the word exo means that it's secreted from the cell, and they're able to form pores in those host cells. So just like alpha toxin for Staph aureus, streptolysin does the same thing. Hyaluronidase, you've heard this before too. This is another enzyme, or exoenzyme, so it's secreted from the cell, and it can degrade hyaluronic acid, which is found in our connective tissues, and that's how it can spread from one site into the bloodstream, for example, or just through the skin. Streptokinase is going to be a molecule that breaks down clots. We talked about how Staph aureus can hide in clots due to coagulase. Streptokinase is opposite, pretty much. So if your body were to wall off Streptococcus pyogenes, it can secrete this enzyme and break through the clot, so it can spread. So this is a spreading. This is also spreading. Strep pyogenes has a nice big capsule, so we know capsules can help those bacteria evade phagocytosis. And it also has something called M protein that is on their cell walls, and it can also help evade phagocytosis. So we can get away from our phagocytes, and we can break through our tissues and spread. So one of the things about Strep pyogenes is that once it breaches the skin barrier through a cut or a wound, it can cause this kind of cellulitis. Okay, so this is what we're looking at in the picture here. Cellulitis is going to be different from those upper, more superficial skin infections because it begins in the dermis, or it can begin in the hypodermis. So pretty deep in the skin. It's going to prevent painful red swelling, pretty nasty infection. And again, strep pyogenes, you could also get that from staph, but we'll talk about cellulitis as a strep pyogenes infection. This is the most severe, probably, infection of strep pyogenes, and this has been sort of sensationalized as flesh-eating bacteria or flesh-eating, I think it's just called the flesh-eating bacteria, but it's flesh-eating bacterial syndrome. It's rare, okay, but you can see how awful it is, and it is potentially fatal. Basically, we will talk about this again with Clostridium. It's Clostridium perfringens, but they are going to be able to infect the skin. They're going to go deeper in the skin. It's going to really eat away at your tissues and kill the cells. So it's going to, so if you're not familiar with the word fascia is connective tissue between the skin and muscles. So just think of it as like a packaging that will separate your skin and muscles. And so the strep pyogenes can get in there and basically infect that. Then they have your proteases, which will basically destroy our host tissue. So it also, nasty guy, inactivates complement. That's part of our innate immune system. And it also prevents our neutrophils from getting there. So it's going to be pretty much unstoppable because of these proteases, unless you have very quick treatment. And the necrotizing, whenever you see the word necrotizing, means that we have death of the tissue, which means that the regenerative cells that are normally in your skin, the regenerative cells that are in your connective tissue are gone because they're dead. And so those areas of skin do not come back. When you see black, this is a very profound sign of necrotizing fasciitis, or any necrotic tissue is going to be black. Once you see necrotic tissue, it's not going to come back, right? So a lot of times, or most all the time, you have to get rid of it. You have to just take it away from your body. So that's going to be called debridement, surgical removal of that dead infected tissue. If it's severe enough, you might even have to amputate the limb so that the bacteria can stop spreading, because we know that it can spread very easily. All right, next one is Pseudomonas aeruginosa. We worked with this guy in lab as well. This is a gram-negative aerobic bacillus. This is common in the world, so it's not something that is harbored on our skin. It can be, but it's also found in water and soil, and as our human skin flora. And normally we think of it as opportunist. It's pretty, for healthy people or people without a wound, our immune system are able to get rid of Pseudomonas, but it's found mostly in wounds or in burn victim wounds where you have open skin and it can cause infection. It also can get into the ear. So this is also known as swimmer's ear. So if you are in water, natural water, for example, that can enter your ear and cause infection. Otitis externa is going to be an outer ear infection. Okay, so if you are unfamiliar, the ear has three parts. There's the outer ear, which contains your auricle or pinna. Then it has a tube, right, that goes into where your eardrum is. And that would be the external canal. And that's where the infection would stay. The barrier from the outer ear to the inner ear is the eardrum or tympanic membrane, and that will stop infection from going forward most of the time. What's interesting about Pseudomonas is it does have a distinct smell, so grape soda or fresh corn tortillas. And another interesting thing about Pseudomonas, it does some of them make this really hallmark blue or green pus. It has a pigment that it makes that it looks that way, and it does help the bacteria obtain iron for growth. So it does have a role. And the last thing about Pseudomonas you want to remember is it does form biofilms. So if you remember from other chapters we read, Pseudomonas biofilms were really prevalent in hospital settings, and it's the one that is hard to get rid of because it does form those biofilms. This is Pseudomonas infection between the toes. You can see that green color. And lastly, we have acne. So acne is going to be, everybody gets acne, and mostly teens, right? 80% of teenagers and young adults. But everybody gets acne once in a while. The adolescents get it due to the hormonal changes that result from puberty, and you overproduce the sebum. Sebum is that oil that is found in our skin, and it can contribute to acne because it's what the bacteria like to eat. So if you look at the picture here, we have a normal, this is the hair follicle. So the hair goes down. The hair follicle is this whole structure here. Every hair follicle is going to have a gland, right? That's called the sebaceous gland, sorry. And that's going to be where your oil is formed, right? We know that if we don't wash our face or don't wash our hair, our skin gets oily, our hair gets oily. So this is the oil. So when under normal circumstances, the pores open, the oil that's made comes out, and the oil coats our skin to keep it lubricated and soft. A whitehead forms when there's an infection in this region, and a blackhead forms when you have oxidation of the pores. So whiteheads are not oxidized yet. You just have the sebum mixed with the inflammatory products such as the fluid and dead cells. And then blackheads. So comedone, you probably not heard this word before. Comedones is basically acne, what we call like pimples, and they can result from these clogged hair follicles. And again, the black spots, blackheads, because of the oxidation process of the lipids in the sebum and with exposure to the air, right? Oxidation means that oxygen comes in and it just turns it black. The most prominent bacteria that will form acne is this guy, Cutibacterium acnes. That's kind of an easy one to remember because you don't really feel cute when you have acne. And yeah, so this is going to consume our sebum and sort of have lots to eat in our hair follicles. So how you typically treat acne is with removal of the skin cells. So you want to sort of hasten or speed up the process of skin cells coming off, and this is really common. You'll see in all those soaps and acne products. You'll also have products, this is a little bit more severe, retinoids that will reduce the inflammation. And then if you are really suffering from acne, they will give you antibiotics, and sometimes the creams will have antibiotics in them. All right, last one here of this, I think, of the skin, we have anthrax. So anthrax is caused by Bacillus anthracis. I failed to mention it up here, but let me draw your attention to the Bacillus anthracis. And this is a bacteria that is found in the soil, very common in the soil. It's a spore former. Anytime that bacteria live in a soil, most of the time, it's a spore former because the soil is kind of a place that's, can dry up, it can become inhospitable. So what normally happens, this bacteria can enter the body several ways. So there's skin infections, which we call cutaneous anthrax. Then there is, if you accidentally eat those spores and they germinate inside of you, that's gastrointestinal anthrax. And then if you inhale the spores, that's called inhalational anthrax. In terms of the skin one, right, it's going to produce an eschar, and you see the black, that's going to be necrotic tissue. So it's a very serious infection, but it can be treated with antibiotics. Untreated, you can have 20% death in patients. So that's a very high number. This is a round, just a nice chart of our anthrax. So we have our, I guess we can start here, right? Oh, let's start here in the environment. So in the environment, we have our spores, and it's very commonly found in animals, farm animals, for example. But for us, right, we can get it three ways. So respirational anthrax, we inhale the spores. Gastrointestinal anthrax, we accidentally eat or drink water contaminated with those spores. And then cutaneous anthrax, it's, wanted to say this, wool sorter's disease is what the nickname is for this, because people who shear sheep, right, the wool is not clean. If you've seen sheep images, right, they're pretty dirty. So people who shear them are going to get nicks and cuts all the time on their skin, and the bacillus that would be from the soil on the sheep would get into the person and cause that cutaneous anthrax. Anthrax is very deadly in these two forms. The respiratory form and the gastrointestinal form are very, very, they're deadly. And they can also cause meningoencephalitis. All these guys can, right? If the bacteria spreads in your body and gets into the brain and covering of your spinal cord, it can cause meningitis, which is very deadly and very serious. All right, so this is an overview of what we've gone through so far. So just know the ones I've starred, and I won't be testing you on the ones that are not starred. Next up is infection of the eye. So we're going to stop here.